Okay, so we talked about mutations in class, and I wanted you to remember that a point mutation, or also called a base substitution, is when you change a single base in the DNA, or you can have an insertion or deletion where you add or delete a single base in the DNA. So for DNA, you can either have a point mutation, or a base substitution it's also called, or you can have an insertion or deletion. Now, at the protein level or the mRNA level, you can have things like a silent mutation. So a silent mutation is when you have a mutation that takes place, like for instance this T here has been changed from the A that it was here, so we went from a GTA to a GTT. However, um, the amino acid has not changed. So there was a mutation, but there was no mutation on the amino acid level, so that's called a silent mutation at the amino acid level. This would be a base substitution at the DNA level. So to sum that up, we would have a base substitution that led to a silent mutation. Now a missense mutation, that's when we change an amino acid. So again, we go from having TAC here to TAA. That goes from a proline coded for it to a threonine coded for it. So we have a point mutation or a base substitution that led to a missense mutation. Now a nonsense mutation, what we change for is we change, instead of a different amino acid, we make a stop codon. So as you can see here, we have TAC, we do a point mutation to TAG. That changes from a tyrosine to a stop codon. That is a nonsense mutation. So this we had a point mutation making a nonsense mutation. Now, an insertion or deletion in the DNA causes at the protein level or the mRNA level a shift in the reading frame. So we call that a frame shift. And you can see here that we have a TA that gets deleted. So that TA is no longer there. So instead of reading AGCGUA, we read AGCGCC, where we have now removed this UA because of the DNA, we have deleted the TA. So that goes from being serine, valine, proline, tyrosine to serine, alanine, leucine, leucine, because now we've changed this codon, we've changed the next codon, and we've changed the next codon because we've deleted two base pairs. So that was a deletion mutation that caused a frame shift, which then caused multiple missense mutations. Okay, so I want to talk about a mutation that is actually beneficial in the population, because we usually talk about mutations as being detrimental. There is a mutation called CCR5 delta 32, and if you'll look back at chapter 6 where we talked about HIV, CCR5 is the co-receptor for HIV. So these people with CCR5 delta 32, they are lacking a functional CCR5. Because they do not have that co-receptor for HIV to enter into their cells, they do show some resistance to some strains of HIV. So it is actually a beneficial mutation. X-rays and UV light. So X-rays break DNA. UV light, which obviously breaking DNA is bad, UV light causes adjacent thymines on the same strand to form covalent bonds. We call that a thymine dimer. So UV light hits these adjacent T's on the same strand of DNA, and that causes them to form a covalent bond. We call that a thymine dimer. To correct a thymine dimer, you must have these cut out. And so you usually do this about 10,000 times a day when you are exposed to sunlight. This is the mechanism, nucleotide excision repair. It takes the thymine dimer that was formed from UV light, and it cuts a region out and it replaces it with non-thymine dimers. So you do this fixing over and over and over every day that you get exposed to UV light. As you get older, this repair system that fixes this becomes less accurate because you've been exposed to more mutations over your lifetime, and because of that, you're more prone to skin cancer as you get older. We're going to skip past replica plating because I just don't think that that's important. We are going to talk about the Ames test. For the Ames test, you take a chemical, and the goal is to see if this chemical causes mutations in your strain of bacteria. So we have a Salmonella strain here whose histidine gene, that gene that makes histidine, has been mutated so that it no longer functions. Histidine is an essential amino acid, so if this strain can't make its own amino acid, it will die. So you must supply it with histidine. So we take this histidine strain, this strain of bacteria that can't make histidine, we give it the nutrients, and we then plate it on an agar that lacks histidine. So without histidine, it cannot grow because it's an essential amino acid that's required. We then take a separate tube where the histidine requiring strain is grown, and we add the mutagen in addition to some nutrients. We then plate this on agar that lacks histidine, and we look for growth. If we see growth on this plate as is shown here, then that tells us that they have had mutations to that His gene where they couldn't make it before, so we call that His minus, and now because this chemical has caused mutation to that gene that has had it to start functioning again so it can make its own histidine, they can now grow on their own. So we say that it's gone from a His minus to a His plus because of mutations, and those mutations are from the chemical. So bacterial gene transfer, we've talked about the ability to do transduction. Now we're going to talk about conjugation. Conjugation is going to involve the formation of a tube structure using the pilus. The way this works is that you have a donor cell that contains a plasmid. That plasmid has some sort of beneficial gene that will be transferred to the other cell which does not contain that plasmid. The cell that has the plasmid, we call that an F plus cell. The cell that does not have the plasmid, we call that an F minus cell. You must have an F plus cell combined with an F minus cell. The way they do this is through the pilus, which creates a tube structure which allows for transfer of genetic material. We then, through the rolling circle of replication that we've already talked about, send a copy of that plasmid across to the other F minus cell. When that F minus cell receives that plasmid, it then becomes from F minus to F plus. That is how you then transfer beneficial genes quickly from one bacterial cell to the next bacterial cell. An operon is a series of genes that work together to make proteins that function in a common pathway. So for the trip operon, the genes here are going to function, these enzymes are going to be functioning to make the enzyme tryptophan. So you have TRPE, TRPD, TRPC, TRPB, and TRPA. These all are enzymes that are involved in the process of making the amino acid tryptophan. So the way this works is that we have a promoter, we have an operator region, and then we have the genes. In the absence of a repressor protein, the RNA polymerase can bind to the promoter, go through the operator region, and make mRNA of these genes, and then you will make the proteins of those genes. That is in the absence of tryptophan. So if this cell is not in the presence of tryptophan, you will then make the enzymes necessary for you to be able to make tryptophan. However, if that cell comes upon media that is rich in tryptophan, it does not need to make its own tryptophan. The tryptophan will bind to the repressor protein, which then allows it to bind to the operator. When that repressor is bound to the operator, that then blocks RNA polymerase from doing transcription. You don't make the mRNA, therefore you do not make the enzymes for making tryptophan, which is a good thing because you don't need it because you have encountered tryptophan in the nutrients around you. So this is called a repressible operon. Now this is in contrast to the lac operon, and in the presence of RNA polymerase and the absence of lactose, the repressor binds to the operator. That blocks RNA polymerase from being able to do transcription on the genes necessary to break down lactose. Lactose is a sugar. So when lactose is present, lactose binds to the repressor, taking it off of the operator. Removing that allows RNA polymerase to then go through and transcribe the genes necessary to break down lactose. So this is called an inducible repressor, because if there is no lactose present, you do not want to be making the genes to break down lactose, because there is no lactose present. If there is lactose present, you do want to be making the genes to break down lactose so that you can use it for a source of energy. Now, cyclic AMP is taking ATP and cyclizing it to make cyclic AMP. The reason why you make cyclic AMP is because there is low levels of ATP, which means there is low levels of glucose because you are not doing glycolysis. So remember, cyclic AMP means low levels of glucose. So, in the absence of cyclic AMP, the cap protein does not bind to the promoter, and transcription can take place at a very, 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 very slow rate. And basically, it's a weak promoter that doesn't bind RNA polymerase very well. Therefore, it doesn't make a lot of mRNA of these genes to break down lactose. So, the only time that you have a strong expression of gene expression is when the cap complex forms. The cap protein binds to cyclic AMP, and then that strongly puts on RNA polymerase to the promoter. The reason why cyclic AMP is present is because you have used up all of your glucose, and remember, the repressor here is not bound, so that means that we are able to do transcription through the genes and make the enzymes necessary to break down lactose. So when the presence of cap complex, if you still have the repressor bound, you cannot do transcription of the genes. So if we have a scenario where the repressor is bound even though there is cyclic AMP here, we cannot have gene transcription.